All right, exceptions to Mendel. Last week we talked about Mendel, who he was, all of his experiments in something called complete dominance. And the pointer, go. So we're going to go past those simple things that Mendel was doing for his experiments. So what he did is he worked with a fairly simple system, an organism, those pea plants. When we're talking about genetics, pea plants are actually pretty simple. Most of their traits are controlled by a single gene. So, we had the color for the flower was either big P, heterozygous, or little p. Just that one gene controlled the flower color. And each of those genes only had two alleles, two P's for that flower color. One happened to be completely dominant over the other. It was an example of complete dominance. If that big P was there, it was going to be purple. If there was no dominant trait, it was going to be white. It was one or the other. Pretty straightforward, no gray areas, no mishmash spectrum. It was just a purple flower or a white flower. It was a round seed or a wrinkly seed. Pretty either this or that, simple, controlled by one gene, and each gene only had two alleles. But unfortunately, genetics is not that simple. Complex organisms like ourselves, other plants, other animals, we just got too much going on. It's not ever going to be that simple black and white. So what we're going to look into are some of these exceptions to the basic complete dominance of Mendelian genetics. So we're going to start off with something called incomplete dominance. So Mendel focused on complete dominance. Now we're going to talk about something that's incomplete in terms of dominance. Incomplete dominance is when a heterozygote shows an intermediate blended phenotype. So we're mixing together the two phenotypes to make a third. So for example, we're going to look at some flower colors again, but a different type of flower carnations. And red flowers are extra dominant. If you have a homozygous dominant genotype, you will have red flowers. And white flowers, just like our pea plants, are recessive. So if we have a homozygous recessive genotype, little r, we'll get white flowers. However, when things get a little bit different, we look at our heterozygotes. And that big R, little r genotype will give us the phenotype of pink. Well, how do we get the pink? Well, we're going to have about half the amount of color. When we mix white and red, we get pink. It's sort of a blend of those two phenotypes. Before I show you the pictures really quick, there is another way that incomplete dominance can be written. The total dominant stays the same, big R. Then things can get a little weird. We can have a different letter for the recessive color. So in this case, WW for white. And then that means our heterozygote has one of each of those letters. I'm going to do my best to stay away from that, focus on the sort of genotypes we're used to with same letters, different capitalizations, but I wanted to put that out there so that if you do see it, whether that is in my class or beyond my class, AP, college, career, whatever, you don't freak out. So here's what it looks like. Here's our homozygous dominant red carnation, big R. Then we have our heterozygous incomplete pink flower, one of each. It could be big R, little r, or it could be R and W, one of each from the red and the white flower. And then our recessive white flower over here could be WW for white or little r, because it is the recessive phenotype. Here's another way to look at it. So we have our true breeding parents, red, white, homozygous dominant, homozygous recessive. Thanks to Mendel, we know that the F1 generation is going to be 100% heterozygous. And if it's a heterozygote, what does that mean about its phenotype? If it's incomplete dominance, we're going to blend together those two alleles. So instead of just red or just white, we're going to mix them together. We end up with pink. So the 100% heterozygous also means 100% pink flowers. We get one allele from red, one allele from white, mix them together, we get that pink. Now, we're going to take our F1 generation, 100% pink, 100% heterozygous. We're going to self-pollinate. But what does that mean for our ratios here? Well, let's look at some of our Punnett squares. Would you look at that? So we put our heterozygous mother on the top, heterozygous father on the side, or vice versa, they're both heterozygous. And this is what we get. We get the same genotypic ratio as we would see in a complete dominant scenario, like with the pea plants. We have one homozygous dominant, we have two heterozygotes, and one homozygous recessive, a 1 to 2 to 1 ratio. With incomplete dominance, our phenotype, or our phenotypic ratio will be the same as our genotypic ratio. So we'll actually get one red out of the two pink out of the two, and one white out of the two. So in this case, our genotype and phenotype, their ratios are going to be the same. If you think back to Mendel, in his complete dominance, it was either purple or pink. Our genotypic ratio and our phenotypic ratio were different. Our phenotype was 3 purple ones and 1 white one. So we're starting to see some differences here between Mendel's complete dominance and this new incomplete dominance. All right, now we're going to move on to a new one called codominance. Codominance. This is when two alleles affect a phenotype equally and separately. So incomplete dominance, we're blending them together. They're going to be one trait. Codominance means that those two individuals are going to be present equally and they're not going to mix. They're going to be separate. What does this mean? Let's work with an example. Humans have a blood type group. You can be A, you can be B, you can be O, or you can be AB. There are 3 alleles, that A, that B, or a recessive O. And then the AB blood type can be codominant. It can have both of those dominant A and B expressed equally and separately. That little I is recessive for O, so you cannot have O-type blood unless you have a homozygous recessive little I. So that part still is the same. But where we're seeing a difference is in these dominant alleles. So let's look at a table to sort of organize this information a little bit better. We have, I always forget what this is called. Blood type is often symbolized by an I with a little superscript of whichever type it is, A or B. That I, we use the I because it stands for, I have it in the speaker notes here, isoagglutination, what is it? Isoagglutinogen, which is just another way to say antigen. So that's why we use an I instead of the just A and B. We have a little superscript to sort of tell us that it is an A or a B because both A and B are dominant. All right, the genotypes here for A and B look just like Mendelian genetics. You can be homozygous dominant for A, so big A, or you could be heterozygous dominant for type A blood, an A, or that recessive little I that is also recessive for type O. So you can have a homozygous dominant or a heterozygote. Same thing for type B blood. You can have a homozygous for B, capital B, or you can be heterozygous. You can have one of those dominant B and a little I that's recessive. Now this is where things get a little bit different. We can see our codominance happening. People with type AB blood. I happen to be one of those weird people who was codominant. When their parents' gametes fertilize into a zygote, the offspring gets one allele for type A and 1 allele for type B. But because they're co-dominant, both of them are going to be expressed equally and separately. So for me, I got an A allele from my mom or dad. I got a B allele from my mom or dad. I don't remember which one was type A and which one was type B, but whoever was type A gave me an A allele. Whoever was type B gave me a B allele. So I am co-dominant for AB blood. And lastly, here we got our little type O blood. This is just like regular Mendelian genetics. In order to have type O blood, you must have a homozygous recessive I to get that gene expression of type O blood. So what does this mean for like blood donations and stuff like that? Let's say me as a type AB blood person, I get in a horrible car crash. Whew, knock on wood, that's not gonna happen. And I'm losing a lot of blood. I need donations. I need a transfusion. I am what's called a universal recipient. I can take any type of blood because I am co-dominant for AB blood. I can take blood that has that A antigen on it from someone with type A. I can accept blood from someone with type B blood because on their alleles, they have a little B antigen. I have both A and B. I can take both A and B blood. I can take AB blood because that's what I am. I can also take type O blood. Type O blood is another special one. If you notice, when we have our recessive little I, there are no letters attached. There are no antigens. This means any blood type can accept type O. There's no little antigen hanging onto each blood cell saying, hey, I'm an A. If you don't have A, we're not going to get along. There's none of those antigens attached to the type O blood. So type O blood is called the universal donor. Anyone can accept type O blood. Again, because it doesn't have that A or B allele attached, the little antigen.
So
we're going to get...
Oh, sorry.
Go ahead.
I was going to say, so that's why type O blood is the most sought out in terms of blood donations.
You are correct. It is the most sought out because it's the most useful. You don't have to worry as much about what the patient's blood type is because if it's type O, they're most likely going to be able to take it. Good point. So we're going to learn a little bit more about blood typing, how that works and stuff when we do our lab. I just wanted to touch a little bit on it so some of it sounds familiar when we do the lab. All right, here's another example, codominance in cows. So we have a totally solid red cow here, and it's going to reproduce with this totally solid white cow here. Red is dominant, white is dominant. But what happens is, because they are co-dominant, the offspring is what's called a roan coloration, a roan cow. Notice how it's patchy. It's got spots and blotches of both red and white hair. If this was incomplete dominance, we would get a solid sort of light red cow. But because it's co-dominance, both of those traits, white and red, are present equally and separately. Can type O take any blood or only other type O? Great question. Type O blood can only accept type O blood because it can't have those little antigen A and B's on there. Good question. All right, next up, weird word. This is called pleiotropy. Pleiotropy. Most of our genes are actually pleiotropic. This is when one gene affects more than one phenotypic trait. So two really obvious examples that I decided to choose were dwarfism and gigantism. They're kind of on opposite sides of the spectrum, but one trait of dwarfism or one trait of gigantism will change lots of different phenotypic characteristics. Someone with a dwarfism gene is going to have small hands, short legs, small torso, short legs. It's going to affect lots of different phenotypic traits. On the other side of the spectrum, we have gigantism. There's one trait for gigantism. If you have that gene, well, guess what? You're going to have long legs, big hands, big head. Everything is going to be big. My favorite giant, person with gigantism, is Andre the Giant. If you ever watch Princess Bride, he is in Princess Bride, which is a wonderful movie that I love. But if you look at Andre the Giant, compared to like an average dude, look how much bigger he is. His arms are big, he has giant hands, his head is bigger, everything is big. That one gene for gigantism affected many different traits or characteristics in his phenotype. Pleiotropy. All right, epistasis. This one is super complicated, so try to stay with me. We're going to look at two different examples. First one up is mice. So coat color in mice is an example of something called epistasis. Epistasis is one gene completely masks another gene. So let's see. Over here we have two mice. They are heterozygous. This is going to be a dihybrid cross. We're measuring two different genes, an A gene and a B gene and a C gene, and there are two alleles for each of those. But it's not going to look exactly like the dihybrid cross we were used to with Mendelian genetics. Let's take a look at all of these little factors here. So wherever you see a C, the C is going to tell us whether or not our mouse is going to have pigment in their fur. Pigment will be dominant. We want our mice want to have color for their fur, whether that's black or brown, but it's more useful if you think about a mouse in the wild to have pigment in their fur. If it's a white mouse, it'll most likely stand out and get eaten. But anyway, pigment is dominant. It has a capital C, meaning if it's homozygous dominant or heterozygous for pigment, it will have a coat color. If, for example, we have a homozygous recessive little c, that means our mouse will not have any pigment. It will have white hair. White is the absence of color. So a white mouse will be albino. It will not have any pigment in it. Once we determine whether or not that mouse has pigment with the C gene, we can look at the B gene. If our mouse has pigment, the more pigment it has, the darker it will be. So we have more pigment in this case will be dominant. So if there is a dominant pigment factor here, a dominant allele, and looking at our genotypes and the way they work, if there is a capital B for that dominant more pigment, we're going to have a black mouse. Brown mice is less pigment. Well, it's a lighter color than black. It won't have as much of that pigment in its fur. And so that is recessive. In order to have a brown mouse, we need to have a homozygous recessive genotype for that B factor. All right, what does this mean? Let's take a look. So here in our first example, we have a capital B and a capital C. So we're going to look at the C's first to determine whether or not our mouse is going to have a color in its coat. Will it have pigment? Well, there is a capital C, so it doesn't matter what the second allele is. That capital C means it's dominant. It will have pigment. Great. We know it'll have pigment. Let's look at the B allele here. It is capital. It is dominant. So again, it doesn't matter what this second one is, if it's uppercase or lowercase. It's going to be black. It's going to have a lot of pigment. So would you look at that? Here we have two black mice. They're going to produce a bunch of black babies. Next example, again, we're going to look at the C first, see if we have pigment. A capital C means dominant. We will have pigment. What type of pigment are we going to have? It is recessive pigment. We have two little b's. That means we're going to have less pigment. It's going to be a brown mouse. So now we have 3 brown mice. And last up, let's look at the C's. Oh, look, we have no pigment. This is going to mask whatever the pigment, the B trait alleles are going to be. So it doesn't matter what the B alleles are because the no pigment completely masks that other gene. So no matter what, if there is a little c in this case of the mice, they'll be white. Now this is where things get a little weird. I know dihybrid crosses, when you have two heterozygous parents, we usually get a 9:3 to 3:1 ratio. But in epistasis, because of the way those genes mask each other, our ratios change for our phenotypes. Now we're going to have a 9:3:4 ratio. You don't need to know that. You don't need to memorize that. Just a little compare and contrast there. Let's look at one more example for epistasis. Labrador retrievers, we all love them. You've seen them. You may even have one. So let's look at our two genes, E and B. E will be our pigment indicator. Does it have pigment? If it has a capital E, pigment is dominant, it will have some sort of pigment. If it does not have pigment, it will be recessive, homozygous, little e. Then if we determine that there is pigment with our dominant E's, we can look at what concentration or how much pigment will be present. Lots of pigment is dominant, so capital B will give us black. Less pigment is recessive. Little b will have to be homozygous recessive for our brown. Let's look at some puppies. All right, so first up we got a no pigment, little e. All right, so we're not going to have any pigment in our fur. This will give us yellow labs. Now, let's look at the B factor. For our labs, when we have a recessive little b, that means less pigment. We get our yellow fur because we have little e. And our little b means we have brown nose, lips, and eye rims. Our other option for no pigment in fur, we have a little e, but let's look at the B factor. We have a dominant B here. So that means our dog will have still yellow fur, because the E, little e, but the pigment in its nose, lips, and eye rims are going to be dark. We have a capital B there. So it'll have black eye rims, nose, and lips. All right, but what if our lab has pigment for its fur? Here we have a capital E. Remember that second one doesn't really matter because it is dominant. We're going to first check out what happens when we have a recessive little b, less pigment. Recessive, we're going to get a chocolate lab. We have brown fur, brown nose, brown lips, brown eye rims. Less pigment for both the fur and the eye rims and lips and noses. But what happens when we have lots of pigment? That dominant capital B. That's where we get our black labs. Black fur, black nose, black lips, black eye rims. So there's sort of a little bit of a spectrum here, but it's all determined by those whether or not there is pigment. Once we determine if there is pigment, how much, based on the dominant and recessive colors there. I know that was really complicated and it's a lot. We will continue to review, repeat, whatever we need to get down epistasis. All right, last little bit here, polygenic inheritance. So some phenotypes are determined by additive effects, 2 or more genes on a single characteristic. These are sort of phenotypes on a spectrum, a continuum, a range. So things like skin color, there aren't just 3 skin colors, we have a whole rainbow. Height, we don't have just 2 heights, tall and short. No, we have a whole range of heights, weights, intelligence, behaviors. All of these things are determined by additive effects, 2 or more genes that affect a single characteristic or trait. Here is just one right off the bat for skin color. There's a whole range. There are many different combinations of skin colors determined by 2 or more genes that we inherit from our parents. Comments, questions, concerns, clarifications?